Welcome, everyone. It's great to see everybody on a beautiful evening. We've had a cold spring, and this is our one day of relief, and now it's going to get cold again. I have a gut instinct that this is going to be a good program. Stick with us. I want to begin by sharing with you the recipients of the Worcester Foundation grants that your gifts make possible. And this is really great. And this year, am I doing the slides? Let's see. We have 4 recipients. And I want to tell you that these are all outstanding projects, but in keeping with tradition, I won't go through them in any technical detail. I want to tell you that Rocco Binder, she's doing a reverse thing. Usually these grants are to help you get a federal grant. She tried to get a federal grant, and the committee that reviewed it had some issues. They always do. And so she's now going to use this Worcester Foundation grant to go back at them, right back at you. And this has to do with the jumping of coronaviruses in Africa. These are the things that have been going on for millions of years, often are benign, but as you all know, recently a jump occurred from a bat that got into the Chinese food market, was not escaped from a laboratory. I think we can be 95% sure now that did not happen. And she's got some collaborators there that have feet on the ground, boots on the ground, and I think that's going to be a very good project. I won't mention the other 2. They're basic science in the tradition of the Worcester Foundation, but Elliot Edmondson has got his hands around a public health crisis, and that is the increasing mental illness of our adolescents. The suicide rate of adolescents, male and female, has doubled in the last 10 years. And in addition, there is a hypothesis that there are children of puberty who have a unique sensitivity to self-image. And this is not chronic anorexia. This is not chronic bipolar depression. These are nuanced responses. And of course, as you have been reading, what has been implicated, of course, is smartphone addiction. And one psychiatrist has made the point that these children who are on their smartphones all the time, you might say, that's the most socializing thing that the world has ever invented. But in fact, further introspection reveals that these children are actually feeling quite isolated. There's an irony there. So Elliot has got the idea that he's going to enroll a group of children who are not affected by this disorder and others who are. He's going to have exclusion criteria. So he doesn't want anybody who's actually depressed all the time. This is an episodic reaction to visual stimuli. After the profiling psychiatrically, he's going to do functional magnetic resonance imaging to see if he can identify clusters of nerve cells that are tuned to this response, either by their hyper or hypoactivity. And by the way, he wrote a beautiful proposal in which he concluded the exclusion criterion of claustrophobia. Why? Because these children, whether the control subjects or the experimental subjects, can't go through the MRI machine. It's a very, very well thought out proposal. And I think it's going to be quite significant. He's got enough patients he's enrolling that he'll have what we call statistical power. That is, the results will have meaning. He will then apply for federal support to expand the study. So thank you very much. And I wanted to mention in particular that Elliot is the Bassick Family Foundation grantee. This is the 10th year that Jack Bassick and his family, who are here, Jack and Susan, why don't you stand up? I don't usually get to boss you around, but would you please stand up? And, oh, sorry, another family member's here. This is really wonderful what you've done for 10 years running, and we're very grateful. And we're grateful to all of you for making these grants possible. Now it's my distinct pleasure to introduce Dean Terry Flott, who is the Celia and Isaac Haydock Professor. He is the Dean, the Provost, and the Deputy Executive Chancellor of our medical school. And as I've said on more than one occasion, I've had deans of various kinds, but to have a dean who is himself a molecular biologist of DNA and working on gene therapy it's a real pleasure to work for you and with you, Dr. Flott.
Well, thank you very much, Thoreau. Thank you all for your support, and thank you all for coming this evening to hear about some of the great scholars that your support has been really sustaining over the years. I have the great responsibility this afternoon of being able to brag about our medical school, or at least give you a brief update as to what's going on in our research enterprise. Oops. So that is not. Or is it just the next slide? Oh, okay. Sorry. Okay, I thought that was going to be her slides coming up. All right. So yeah, so the research enterprise here at UMass Chan continues to grow year by year. This is a slide we update every year that shows our total awards each year. And you can see that while the state support has been relatively stable, a little bit under 50 million over all of these years, that our research has been on a steady trajectory. There are two humps in the curve. The first one of those was from the ERA funding in the Obama administration, for which that was the famous shovel-ready projects, for which we had some additional large grants. And then the second one was COVID-related grant funding, particularly this single $123 million grant that went to David McManus for the RADx grant to test to evaluate point-of-care testing devices. But if you look at the underlying trend, it continues right along and has exceeded the rate of growth of NIH funding overall. So just to quantify some of that, the looking now just at NIH funding, that was total funding on the other slide, 322 million. It's grown from NIH funding from 118 to well over 200 million. And it continues to grow each year. We expect it to grow even further next year. One way that we look at this is department by department. So what this survey nationally called the Blue Ridge survey from the Blue Ridge Institute for Medical Research takes data directly extracted from the NIH reporter, and they evaluate it so that all departments of a certain type compared to similar departments around the country are rated against each other, and then you end up coming out with an overall rank, which you can see right here. So we let each department, our basic science departments no longer fall into the classic categories they used to be at most medical schools, named by the names of the courses that medical students took before they went to the clinic. But the two departments here, program in molecular medicine and molecular cell and cancer biology, both self-classified as cell biology departments. Their combined efforts, and I think reflective of our cell and molecular biology prowess, is in fact well ahead as the number one in the United States. Our dermatology department, a little more consistency there. This has been a very rapid rise under the leadership of our new chair, John Harris, who is an MD PhD graduate of UMass Chan Medical School. It had a very small research enterprise before he took over his chair 3 or 4 years ago, and now it's almost to the top 10. Emergency medicine has been in the top 10 relative to other emergency medicine departments for many years. We have 2 departments like self-classifying genetics, including the Gene Therapy Center, which is where my lab is, and they rank 12th. Now you might say, how does one with $21 million rank lower than one with $6 million? Again, this compares like to like across all U.S. medical schools. And you see the ranks all the way going down here. So we're very proud of all of the accomplishments of all of these departments. And really, I think anytime we see any of these that are ranking in the top 30s, we're seeing some exceptional growth relative to peers, because there are 155 medical schools now. So that's just by the numbers. The big update in research is, yes, that building is open. It was on transferred. We received the official certificate of occupancy over this past weekend, and yesterday lab equipment began moving in. Actually, it started with the laboratory of our new chair of neurobiology, Dr. Danny Winder, who's just moved from Vanderbilt. And so he got to be the first one to move in because we didn't have to load up his lab from another building. But this building will really improve the space resource for really the entire basic science research enterprise. By that, I mean, if you look at how it's laid out, this is what we call the stacking diagram. The program in molecular medicine is finally, after I'm trying to think, something like 41 years in existence, moving from 2 biotech across the street, we're finally going to let them into the main campus. And no, it's one of our most distinguished departments. That was the department that Craig Mello was in when he won the Nobel Prize. An important feature for molecular medicine is there is an adaptation for a state-of-the-art metabolomics core, which consists of, among other things, 8 mass spectroscopy instruments in one location. So these floors are equipped with extra power and cooling capability. So what's happening is we're bringing the neurology department with the Wellstone Center and the Gene Therapy Center from the Sherman building over to the new research building. They will both get more space, or they will all 3 get more space in that transaction, as will some other departments that have small research enterprises that are kind of subsumed under neurology, like psychiatry and neurosurgery. We're moving the neurobiology department from Lazar building over into the new research building, and they also get more space. So each of the programs moving in will have space for expansion, but as you can see, we then make more space both in the Sherman building, where we're going to have further expansion in departments like the RNA Therapeutics Institute and the Systems Biology Department, the Department of Systems Biology, as well as in the Lazar building, where we're going to expand the space for dermatology and for the innate immunity division in the Department of Medicine, which are 2 of our most outstanding immunology-based research enterprises. So this will lift all boats, as they say. The design inside this building, we hope to have it open for tours soon. It's not quite in that state yet because we're prioritizing moving the equipment in, but it is organized. It is in a form of the open lab concept that we've had in our other research buildings, where researchers don't have hard walls between different labs. Although this one is more in square block configuration, something that the lab designer that we've worked with for many years, Josh Meyer, calls a lab neighborhood, so that one can actually see not just down along a series of benches, but across a sort of a 3-dimensional space. It will also have, as all of our science buildings, we try to have spaces for informal exchanges about science and other things. Each of the floors has a space like this one, which you can kind of tell is on the corner of the building facing the ambulatory care center over there, which would be that'd be the southeast corner of that building. And these have beautiful views. You can't see it, but there's a coffee bar back to the left of this photo. And the chat over coffee between scientists running into each other informally in the hallway has always historically been a very creative moment, and we hope to recapture that. The actual ribbon cutting for the building itself will occur Friday, June 7th. So that is at the end of the week of our commencement starts the week formally, if you call Sunday the start of the week, on June 2nd, and we end on June 7th with this ribbon cutting ceremony at 11 a.m. And it will happen in a location right outside the building, and then the trustees and our guest speakers will be able to go through. We're very pleased that Dr. Tedros Ghebreyesus has accepted the Chancellor's invitation to come be our keynote speaker. As the Director General of the World Health Organization, I think he's developed a great reputation for his vision and his high principles in the implementation of world healthcare strategies. Well, I wanted to take just a little sidelight to congratulate our newest member of the National Academy of Sciences, Dr. Celia Schiffer, who I believe is here. Stand, Celia, so I can see you. Congratulations to you. Dr. Schiffer, I know you've heard from her before. She is really our scientific leader in structural biology. She's an acclaimed biochemist and scientist, the relatively recently started chair of the Department of Biochemistry and Molecular Biotechnology, and to be acknowledged and recognized as a member of the National Academy of Sciences is very fitting, and we are thrilled for you. Just for those of you who might have had trouble keeping track, we went ahead and listed all of our current and past members of the National Academy of Sciences and of the National Academy of Medicine, which was formerly called the IOM. And you can see, amazingly here, we now have 9 current members of the National Academy of Sciences and 2 past members, or 2, as you all know, tragically, we lost Michael Green. Melissa Moore went on to great success as the Chief Scientific Officer for Platform Technology at Moderna, and got in there before they launched on this vaccine campaign. So she was really at the center of the COVID vaccine development and discovery. But we're also very thrilled for all of these current members that we have. This school has catapulted forward in this way. I believe that when I arrived here, we had Craig Mello and Victor Ambrose, and that was all. Roger Davis at that point was an inductee in the Royal Society from Britain, but had not been recognized for this country yet. But it's wonderful to see all of our wonderful faculty recognized. So with that, I'm going to end my comments about the overview of research, and I think go right into unless Dr. Peterson has anything else to comment, go right into my introduction of our keynote speaker for this evening. So it is my pleasure to introduce to you essentially your endowed chair, the Worcester Foundation for Biomedical Research Chair, who is also the chair of the Department of Microbiology and Physiological Systems, the founding director of the Center for Microbiome Research, which has evolved into this program in microbiome dynamics. And of course, I'm speaking of Professor Beth McCormick. So just a little background, Dr. McCormick is a world-renowned microbiologist and GI physiologist who over the years has studied many forms of interaction between gut microbes and the GI tract. She's had a special focus on cross-signaling and metabolomic interactions, and particularly on multi-drug transporters in the GI tract. She also co-founded Adiso Therapeutics, a biotechnology company based in part on her technology, and that company is advancing a first-in-class molecule now into phase 2A clinical trials for the treatment of ulcerative colitis, one of the really difficult forms of inflammatory bowel disease. Her work continues to identify novel ways in which microbes interact with the mucosal surfaces. She's published over 150 original research articles in this field. She serves as an advisor to governments and multinational agencies and is the recipient of many awards and commendations, including being elected as a fellow of the American Academy of Microbiology, an American Gastroenterology Association Mentor of the Year Award, a distinguished scientist to the American Physiological Society. She's also, in her spare time, editor-in-chief of the journal Gut Microbes and an elected council member of the American Gastroenterology Association, and finally, an elected member of the governing board of the American Physiological Society, American Physiology Society. I will say that Beth has also, on a less formal note, been an outstanding leader at our institution, advocate for science, and one of the people who, along with the rest of her team that she will tell you about, stepped up during the COVID pandemic. We all are familiar with the fact that a little ways into the COVID pandemic, we were suffering one wave after another of different variants coming through. And we found out that the Massachusetts Department of Public Health, in surveying the arrival and spread of different variants, was only sampling in Boston because they seem to think that Boston was the same as Massachusetts, and that we didn't necessarily agree with that. So Beth stepped up to this challenge of not having any genomic data from the SARS-CoV-2 virus from Central Massachusetts, and in an important collaboration with colleagues at UMass Memorial and in the biorepository and other ways, got with her team generated updated data on a regular basis to so that we could have the availability of knowing where the variants were in Central Mass and even within which areas within the city of Worcester. So that's a public service that I don't know has been adequately recognized, Dr. McCormick, but we certainly do appreciate your doing that. And now we'll ask you to come up and tell us a little bit more about the microbiome and your own research. Thank you for joining us.
I thought I could do this without my reading glasses, but I don't think that's so here we go. All right, Dean Flott, thank you for that very generous introduction. All right, so for the next 20 minutes or so, I think my charge is to introduce you to your gut and the microbes that live in it, and give you perhaps some perspectives that you didn't otherwise think about. So okay, off we go. So I want to begin with posing to the audience here, are we more microbial than human? All right. And I pose that as a question, and we're going to come back around to that at the end, because our bacterial cells outnumber our own body cells by a factor of 10 to 1. In addition to that, these microbes comprise up to 4 to 6 pounds of your own body mass. And when you think of the genomic potential, our own human microbiome outcompetes our human genomic potential by about a factor of 40. Okay. And so where do these microorganisms live? Well, they happen to live in and on us. And I just outlined here some of the sites that you probably didn't know that you had microorganisms inhabiting. Your skin and mucous membranes, your upper respiratory tract, the gastrointestinal tract, of course. You also find them in your reproductive areas, in your ear canals, in your eyelids. So they're all over us. And if you take a look at the different communities of these microbes, and I don't expect you to understand the phyla of these organisms, just follow the colors. And I think you can appreciate that the areas that these organisms habitate in our bodies, in us and on us, are different. So if you compare the large intestine even to the small intestine, you see different microbial communities, which is different from what you find in your ear canal, in your mouth, and so on. And so I'm particularly interested in the gut physiology, and it so happens that 95% of our bacteria are located in our gastrointestinal tract. And so, so this is what we're going to be talking about for the rest of the talk. And so if you take a look at these intestinal microbiota that inhabit your intestinal tracts, it is a community of about 100 trillion microorganisms. And there is debate in the field as far as the number of species. Some come in at about 1,500 species. Other projections are as high as 10,000 species. But the take-home message, if you take a look at the anatomy of your alimentary tracts, going from your stomach all the way down to your colon, you see that these organisms not only exist in different numbers, but they exist in different communities. And so these organisms are not there by happenstance. They do impart very important functions for us. And I'm going to go through a couple of these, not in a very down in the weeds, but just to give you some perspective that they do provide protective functions. There is a concept in the field called colonization resistance. And so you have all these organisms that are inhabiting your GI tract, and they're there to kind of keep out the pathogen intruders as they come in. They also produce a lot of antimicrobial factors, such as bactericidins as well as lactic acids. And so these commensal organisms really do their role in terms of also structural functions. So barrier fortification. So our intestinal GI tract needs to make sure that we're not letting these intruders in. In addition to that, it's a place in which educates our immune system. And so there's important immune system development that also happens in communication with the microbes in our gut. And finally, there are important metabolic functions. They produce short-chain fatty acids that feed back onto as an energy source for the intestine. Many of you probably don't even know that they produce important vitamins such as vitamin K, biotin, and folate. And also they are important in ion absorption. So they impart all of these important functions. So when do we become colonized with our gut microbes? It turns out that colonization of the GI tract begins immediately after birth. And the colonization pattern is affected by the mode and type of birth delivery, the initial diet, whether it be if these babies are breastfed or whether they are formula fed, and also the geographic location globally will affect the colonization pattern. Complete colonization occurs around 18 to 24 months. But we don't, exist with the microbiota that we were born with. So along the way, as we get older, we adapt to our GI communities. And again, just follow the color here. And these transitions happen for a variety of reasons. When you are a baby, again, whether you're breastfed or formula fed will impact the communities. The transition from formula food and breastfed into solid food also provides another inflection point of these communities to come in. And also as we become into, adults, and there's antibiotic treatments and drugs. And so all of this occurs throughout our life. And so as you can see here, the communities that we are born with undergo adaptations. And these adaptations can be good adaptations, or they can create problems, which I will sort of shed a light on. But, in terms of our microbiome communities, even though we're healthy, it turns out that my microbiome is not the same as Thuru's microbiome. And that's because the number and amount and the many different microbes can vary greatly from person to person, even though we are healthy. And that is because there are these environmental influences that I alluded to on the prior slide, where our lifestyles play an important role, whether or not we take, on courses of antibiotics, our diet, our hygiene. And if we are, good in all of those things, we have a nice normal microbiota. But if there's influences that perturbate the system and cause a state called dysbiosis, And then this state becomes an unbalanced state. And the reason why that is important because not only does 95% of our bacteria are located in the GI tract, but it's also where 70% of our immune system is tasked with interacting with our gut. And so as you can see here on the left side here, if we have balance where we have rich and bacterial communities, they communicate in a very healthy feed-forward way to impart health. But if there's perturbations in the system, and if these perturbations become sustained, then what happens is that there's miswiring in the balance that gets our immune system kind of out of whack, and we can create some pathologies, autoimmune diseases and such. And so what we are trying to do in terms of the grand challenges of understanding the microbiome is to understand it in terms of implications for human health and disease. So what is, that normal state of gut homeostasis? How do we fall out of that healthy state to this dysbiotic state? And when we are in that dysbiotic state, how can we restore that back to health? And the reason why that's important is because these dysbiotic states associate or correlate with a number of diseases. Inflammatory bowel disease is one of my favorite diseases. We study Alzheimer's disease within the center. And there's also obesity, type 2 diabetes. So whatever disease type you can think about, there is this association with the gut. So if we have, a situation where we fall into dysbiosis, can we modulate our microbiota to bring us back to a more healthy state? And so that was the thesis for the starting of the Center for Microbiome Research. So our mission is to define the interactions between microbes and their environments, between the microbiome and the health of the host, as well as the factors that maintain or promote the healthy function of these ecological niches. And so our model really is to advance discovery into practice. And what I mean by that is by going from learning from the bench, going to the bedside, and coming back to the bench. And so what we do is we take an integrated approach to this by integrating clinical, experimental, or biological, as well as computational approaches to understand how the gut microbes interact with us with the purpose of developing perhaps microbiome targeting therapeutics to prevent and even treat disease. And so this is us, and I want the members of the team to stand up. So my greatest pleasure in my work life is to come to work with these folks. So, and so this is Doctors Evan Bradley. Evan, please stand up. Dr. Vani Bushi. Dr. Ana Maldonado, who actually co-founded the center with me. Dr. Doyle Ward, who is our Director of Operations. And Dr. John Heron. So this team that we have here assembled today have posters up in the reception area. So afterward, you can mix and mingle. I want to just give you just a very high level of what they do. So Evan studies the impact of the microbiota at its intersection with infectious disease. Vani Bushi uses artificial intelligence and predictive modeling to understand things that we can't understand when we're working experimentally. Ana is our diet guru. She uses food and diet as therapy to help modulate the microbiota. Doyle works with pathogen genomics and surveillance, and that study that Dean Flott was very gracious in sharing was actually done under his astute direction. And then John studies the gut-brain axis, and so he has a very nice poster set up that talks about not only the gut-brain axis, but puts it in context of Alzheimer's disease and studies that we have ongoing at this institution to study it more in an in-person setting. So I'm going to share the rest of the talk by telling you about my work. And so what I have had a long-standing interest in and studying is what goes wrong during gastrointestinal disease. And I'm kind of agnostic to what induces inflammatory diarrhea. And so it could be infectious organisms like Salmonella typhimurium, or more chronic idiopathic illness such as inflammatory bowel disease. And in case you're wondering, the chances of you getting salmonella by putting your finger in the cookie batter is 1 in 10,000. So, okay. All right, so this is what our intestinal mucosal surface looks like when we're healthy. How many of you have had a colonoscopy? Okay. So this is what the mucosal surface looks like healthy. And if you were to take a biopsy and look at the cell architecture of this, to the right here is this histopathology section. So you can see nice architectural features here. So what happens during disease is very profound. And you can just see, and this is a patient suffering with ulcerative colitis. The wall gets a little thicker. It gets, these areas here are hemorrhaging red blood cells. And these white patches here are, in fact, crypt abscesses and pus. You can see here, I'm not going to go into the high-level details, but there's something dramatically wrong with the histopathology phenotype here. And one of the things that struck me when I was a postdoc fellow is if you take a look at the intestinal crypts during health, this is what they look like. But during disease, you see all these purple dots that infiltrate into the crypts. And these are our white blood cells. And it turns out that the numbers of white blood cells that infiltrate into the crypts correlate with disease severity and also lead to the clinical manifestations of the disease. So I have had a longstanding interest in understanding how these white blood cells get into the tissue. And if we could figure out that process, we could intervene medically and prevent that step from happening. So we have used salmonella as a model for this because salmonella parallels ulcerative colitis clinically, radiologically, and histopathologically. And so salmonella is a pro-inflammatory trigger. It comes into the GI tract, interacts with the intestinal epithelial cells, and promote this really interesting network communication of providing inputs into the epithelial cells that communicate with the immune cells. And so we need to have this in order because if we don't have these white blood cells come into the tissue, they can't help us fight the disease, okay? And so this is probably my most humbling slide because it sort of like encapsulates about 20 years of my life in this one slide. But this is what we have learned about salmonella and its ability to induce this white blood cell infiltration. So the organism comes in to the GI tract. It has literally this needle-like syringe that ejects effector proteins into the host cell. These effector proteins coordinate the ability of the pathogen to enter as well as to promote this pro-inflammatory event. And it impinges on signal transduction pathways that are called MAP kinases to induce this. This is NF-kappa B signaling to rev up a pro-inflammatory event. Not only does that happen, but these salmonella have these structures called flagellin, and they transpose across the epithelium and interact with a receptor on the basolateral surface that also promotes these pro-inflammatory molecules. And this is an example, a key one here is produced called IL-8. That is sort of a sensor, sort of a bakery sensor. So if you're walking across a bakery and you smell bread, IL-8 is that sense, and it gets neutrophils, and the neutrophils smell that, and they come right up into the subepithelium. Interesting, what we found is that this is a two-hit, and so salmonella induces another effector protein that turns on a pathway that induces a very highly potent neutrophil chemoattractant that is then effluxed out the apical surface. This forms a gradient, and then the neutrophils sense that gradient and are sniffed to the site of infection. And so these white blood cells will, in a balanced situation, control this inflammatory event. But what happens is that during disease, this whole process becomes unbalanced, and you can't resolve it. And so this is what happens in patients with Crohn's disease and ulcerative colitis. You see here this ring of this efflux transporter on the apical surface. And that means it's promoting this efflux of this very potent neutrophil chemoattractant that just keeps this inflammatory event amplified. And so in studying this, we wondered, what is it regulated naturally? So how to resolve this? And so by informed sort of thinking around this, we wondered whether or not if there was another transporter that effluxed out another immunomodulator. And it turns out there is. And so this is other efflux transporter that sits on the surface of the epithelium that effluxes out endocannabinoids, that puts the brake on the immune system and quells down the neutrophils to be to the site of infection. And why this is so remarkable is because these transporters are very important for us. They impart very important physiological functions. So think of them as our natural waste removal systems. They remove toxins from our cells, they eliminate drugs. And in this context, you may know them in terms of multi-drug resistance transporters. But these decorate our intestinal surface. And the remarkable thing about what we found is that these efflux transporters are actually effluxing out immunomodulators that are controlling the balance between pro-inflammation and anti-inflammation. And so the signals that they get from here, so this is in a homeostatic state. So you have P-glycoprotein effluxing out these endocannabinoids that keep everything nice and quiet and suppressed. And then in a pro-inflammatory state, they are stimulated to switch their presentation. P-glycoprotein collapses, expression of MRP2 goes up, and you get this infiltration event happening because now the MRP2 transporter is effluxing out this highly potent neutrophil chemoattractant. The other remarkable thing is that we're finding that these efflux transporters integrate signals from our gut microbes to govern this innate immune response. So as our balanced microbiota during an inflammatory event comes unbalanced, is where we see this nice correlative reciprocal regulation of these efflux transporters. And so what we're doing in my lab is we're trying to understand who these microbes are, what they are secreting, how they communicate within the cell and control gene regulation. And we want to be able to understand that, because if we understand what happens if you fall out of balance to an unbalanced state, can we then restore it back to a more healthy state? And so as a demonstration of some success measures in this, so we have been working on therapeutically understanding how we can correct the balance. And so in this scenario here, I told you that when patients with ulcerative colitis have disease, they have this really high density of these white blood cells that are relevant for the clinical manifestations. So our question is, how can we restore the balance and bring these people back to a healthy state? And so work done in collaboration with my longstanding colleague, Randy Mersney, we have designed first-in-class therapeutics to basically plug up the efflux transporter, prevent it from secreting that very highly potent white blood cell chemoattractant. And when we do this, we can prevent disruption of that epithelial barrier. We can overall reduce intestinal inflammation, and overall, there is less intestinal dysfunction. And so this was the launch of the company that Dean Flott alluded to, and we have been working to get this into the clinic. It turns out this is a first-in-class small molecule. It does block neutrophil activity. And one of the things that about this drug is that there's no systemic exposure. So this is an orally delivered drug. It preserves the protective host immunity. And so our hope is that this can be an oral delivery drug that you just take once a day. For any organic chemistry brewers out there, maybe Celia, you can appreciate the structure of it. And so this is our lead candidate that's working through the clinics. We have had successfully gone through in terms of a phase 1. This drug was found to be safe, well tolerated, and also restricted to the gut, as I told you, with limited exposure and no systemic immune suppression. We also have other drugs in our pipeline that are targeted to other immune aspects and working our way into respiratory infections. We are also have a portfolio of single-strain live biotherapeutics that a couple of them are phase 2 ready, and a couple of them are in our IND enabling and discovery pipelines. So in summary, I hope that I have impressed upon you that we have used a pro-inflammatory mechanism using Salmonella as a model to dissect mechanistically what happens during pro-inflammatory responses. In addition to that, we have been studying mechanisms that resolve inflammation or counteract or balance this host inflammatory signals that we have been discovering in terms of natural resolution, but really led to therapeutic intervention strategies around targeting ulcerative colitis. And now we are expanding our studies out to what drives pro versus anti-inflammatory signals. And primarily, we're looking at the microbiome and host microbiome and immune interactions. So I'm going to wrap this up by asking you, are we more microbial than human?
We are now.
We are now. And finally, last but not least, acknowledging all the people that make this work possible. And I'll end there. Thank you very much.
So we'd love to open this up for questions. We need a walk. I'll
What am I
walk
going to
now.
walk?
I'll be a runner.
Okay.
First, I have to.
There we go.
I'll lead with a question.
Okay.
The population dynamics of the microbiome, whether it's 500 species or 10,000 species, must respond to your Salmonella model. And I'm just wondering, are there differential sensitivities in different species of bacteria? And is there a recovery phase so that you get back to the population structure that was there in the first place? I know that's not the focus, but the evolutionary biology, so to speak, of this recovery phase must be very fascinating.
Yeah, so that's a simple question in principle, but hard to unpack. Yeah. So when we are doing these studies in mice, I talked about the concept of colonization resistance. And so when we were to give the organism to mice, it won't colonize. And so what we have to do is feed the animals an antibiotic to deplete their microbiota. And then Salmonella is able to colonize and do its thing. And I think that's what happens in humans as well, because there's a critical mass of organisms to kind of go over a threshold to defend, and invade inside the intestinal tract.
Sure. Thank you. Great.
But one more follow-up thing that I like to stress is that the Salmonella induces an inflammatory event, and we know that the community structure changes in a way that promotes the infection process.
Some of you have been members long enough that you may remember that at one meeting I showed a slide of me in a hospital in Hungary with a Salmonella case that was quite acute, and it was a TB sanitarium that had been converted. And some of you gasped when you saw me, and I regret that I didn't bring it tonight so you could gasp again. It's pretty bad stuff. You can eat the batter, but be careful of an egg. It doesn't look quite right when you break that shell. That's where it lurks. Yes, please.
Just for people who don't know so many things, how can one keep a healthy gut? you know.
How can one keep a healthy gut? So I'm going to have Anna. So I'm going to have some of my members of the. So Anna is our diet.
Anna, help us.
So Anna, why don't you answer that question?
Well, there's not a okay. There is not a perfect diet, so that's the bad news. But we are trying to understand how diet can affect the microbiota and trying to see if we can have a diet that can ameliorate some of these bacteria that are not beneficial and produce inflammation, etc. And I think you stopped my mic poster. You can see that because diet is very culturally attached and it's a behavior. You guys go to the supermarket, go to the same aisles, you cook kind of the same food, and you have like 3 recipes, etc. So how to make those changes gradually? It's a behavior problem. That's how we see it. So we're trying to help patients with those diseases to engage in a behavior that is healthier for them, and then trying to understand actually what's better. Is it carrots? Is it tomatoes? It's just 2 cups or 1 cup, and you try to make the right changes depending on what you eat. But I'm a firm believer that you can have a healthy microbiome if you eat well. And that means different things for different people, and that's what we're trying to understand.
That's maybe related to Anna, so I'll keep it there. Connection between the microbiome and the trendy weight loss drugs that are out there, Ozempic, et cetera. Has any analysis been done on how those things are affecting our microbiome?
I'm sure somebody's doing that.
Yeah.
But nothing's been published on that yet.
And we had the Herbalife Ponzi scheme, which was one of the biggest short sell things on the stock market. So we have to be careful of soothsayers and promisers. Yes. Kim.
Is there a way to determine whether or not you have inflammation before it develops into like IBS or something? Are there any tests that you can take? Is it just you don't feel good? How can you find out you have something wrong before you end up being symptomatic of something?
I think that's one of the grand challenges in the GI field is to identify biomarkers. So far, nothing has bubbled to the surface that is enlightening us in that regard. I will say, though, it's true. So anybody who has a foodborne intestinal illness, such as your Hungarian issue, it predisposes you 20% higher risk of developing more idiopathic chronic conditions. So that's one thing. So there is a huge effort now being done within the microbiome community to figure out whether or not there's signatures or that we can to identify that provide that warning sign of a predisposition, but nothing has been developed yet.
And in terms of like Alzheimer's, if it's in the family, is there a way to test anything in the gut to see if you might also develop that at some point?
Dr. Heron is our Alzheimer's resident expert. I'll let him handle that question.
Yeah.
Sure.
Oh, sorry. I would give.
You don't need this problem.
Makes you sound, yeah, it looks sounds good then. Within the gut, that's what we're working on. We're working on trying to find and understand mechanisms that link with Alzheimer's disease. To step back a sec, the biomarker field in Alzheimer's disease has exploded within the last year to 2 years with different proteomic tests that are being done now to tell you risks of it, and the monoclonal antibody treatments that have been coming out, which we still don't, the jury's still out if it actually has any real long-term impacts for. So there's testing, but then we don't quite know what to do with it. And then there's treatments that we don't quite know how they work. We're working on the gut-brain axis to look at signals and seeing if we can come up with adjunct therapies to help people, because it's not a smoking gun within the microbiome, but there are coming back to inflammation and other mechanisms that are more pronounced in Alzheimer's patients than not. And especially if you look at, just because you have Alzheimer's disease doesn't mean your cognition goes down in a year. It can take a long time. So that's the nuances we're trying to figure out, where the gut microbiome fits in more with trajectory and how their cognition progresses over time.
So should people stop at CVS on the way home and get probiotics?
No.
That's my point about herbal life. Terry.
Yeah, I had one question that I hopefully won't tie in too much with Dr. Makel over there, but I wondered if, about the potential connection between, one classic example, I guess, is that I believe that patients with ulcerative colitis, if they don't have a colectomy, are at higher risk to develop cancers. And I guess the question is there a pro or anti-carcinogenic microbiome relative to something like colon cancer?
Yeah, I mean, I think there's a large literature now that I think does support that. But it's just, it's complicated by the microbiota field itself, because there's these organisms called Fusobacterium that can be signatures of colorectal cancers. But within that genus, there's many subspecies. And so where are they at? What are they doing? And so I think in terms of the contextualized elements of that, I think we're getting a little bit more resolution on those communities in terms of making those correlations. But there's also nuances in terms of the staging of the cancers and so and how they're going to be responding to the drugs as well. Because these microorganisms also metabolize drugs, and they're interfering with the ability of the patients to respond sometimes to their treatment.
Well, this might be John.
One quick question. With the prevalence of antibiotics, not only in the raising of some of our foods, but also obviously from a therapeutic standpoint, how does that impact the things in our gut that are, where you killed some of those in some of your tests, and I didn't know what the prevalence of that would be, as a negative.
Yeah, no, I think there's, books that have been written on this, subject area. Martin Blaser has a book called The Disappearing Microbes, and that's really all about the overuse of antibiotics for the last 50 years. And so what if we do, I was on a course of antibiotics for an abscess that I had, which, I was appalled that the doctor would even suggest that I take an antibiotic, because what's it going to do to my GI tract? But I think, so when you do take it, it's going to, get rid of some of your organisms that exist there. And when it becomes repopulated, it's not going to return to the original state prior to. But there's a lot of redundancy in our communities that exist. And so we are working on the microbial dynamics of these communities, because if you, deplete certain ones, others come in and sort of take the role. So short-term antibiotics is fine. It's the long, courses. And I think the jury is still out in terms of, cause-effect relationships of some of these things. And so that's really the next generation of the microbial sciences, is to go from correlation to cause-effect.
Good question. The dental field in particular, there's certain procedures where you have to be on an antibiotic an hour before the procedure. So, but Blaser, actually, that book has been a sensation, because there are a lot of parents who are shocked when a 13-year-old girl is getting a wisdom tooth removed, and the antibiotic treatment is heavy-handed and probably heavier than it needs to be. This might be a good time to segue, first of all, to thank Beth again for a terrific presentation.
Thank you.
And there are two treats that await us: refreshments and posters. And I would encourage you to enjoy both. Thank you so much. Thank you.